Hello, Dr. Cisse covering anatomy and physiology. Today we'll talk about the nervous system. We're going to do an exam review, question 1 to 20. Here you see the picture of the nervous system on the right side here, the brain and the spinal cord. Inside the brain and the spinal cord, you see interneurons. They're confined to CNS. That means they're in the CNS. Then you see in the PNS, in the peripheral nervous system, you see the sensory or afferent neurons, and then the motor efferent neurons. The difference here, you see one, which is sensory here, is receiving information to give to the brain or spinal cord that will process and give the command through a motor neuron. That's what we'll talk about in general today. Let's talk about the overview of this lesson. This is the exam review on the nervous system, right? So we covered the nervous system in general, the spinal cord, the brain, the autonomic nervous system, and the spatial senses, right? We all know that the human nervous system is very complex. You can see, you can smell, you can hear, you can mix the ideas and stuff. So the goal of the nervous system, the role of the nervous system is what is allow us to be informed about what's out there so that we can respond accordingly, right? So in this today, I'll talk about the structure of the nervous system. I'll also talk about the action potential, how it's generated, and I'll talk about the difference between the brain and spinal cord. But looking at the picture here, you can see CNS in blue, brain and spinal cord, PNS in yellow, all the nerves that come out of the brain and out of the spinal cord. And then you see ganglia, which are cell bodies of neurons in the peripheral nervous system. And here you see nerve fibers. Now, what are the functions of the nervous system? Why do you need your nervous system? It's for you to be informed through a sensory neuron, process in interneurons, which is gonna be what? In the brain and spinal cord, and respond to the situation through your motor neurons to do something. Now, why do we need to do that? Now, without our nervous system, we will not know how things are going on in the world. We see with our eyes, which is the part of the nervous system. We can smell with our nose, which is part of the nervous system. We can hear with your ear, nervous system. We can taste with our tongue, gustatory system. And then we can feel on our skin, general feeling, temperature, if you're cold or hot. All those are done for us to be informed about something, about the taste of the food, the color of the shoes or something like that. And then we can make proper decision. The nervous system is for information, okay? Now, what do the nervous system and brain have in common? Now, both the nervous system and brain are part of the CNS, the central nervous system, right? Now, what else? They're both covered by meninges. And we have 3 layers of meninges. The very top one you see on the brain, it's the same thing you see on the spinal cord. It's called the dura mater. Then the second layer you see on the spinal cord is the same thing you see on the brain, arachnoid mater. And then the third one, the very thin one on top of the spinal cord of the brain, is the pia mater. So the meninges are the same. Both of them are part of the central nervous system. Now, look at the gray matter and the white matter. The very top of the brain is gray, but the inside of the spinal cord is the one that's gray. So that means the gray and white matter are reversed in the brain and spinal cord. Now, if you think about the neurons also, both of them will have interneurons. Now, the nerves that come from the brain are called cranial nerves, but the nerves that come out of the spinal cord are called spinal nerves. We have 31 pairs, meaning on each side, one comes 31 on each side. And then on the spinal cord, 31 pairs of nerve, right? Spinal nerves. But on the brain, we have 12 pairs of cranial nerves. So the brain and spinal cord have so many things in common because they're both the same system. So the spinal cord will be just what? The extension of the brain. They both have cerebrospinal fluid that is secreted, it's going to be running through the brain. Through the spinal cord, it will go through the central canal, but in the brain, it will go through the ventricles, right? And between the meninges. For example, you see subdural space or subarachnoid space here. You see that? So you see some kind of cerebrospinal fluid running through the brain, around the spinal cord to give them nutrient and to take waste. Now, how is the nervous system divided? The nervous system is divided into the central nervous system, made of the brain and spinal cord, and the peripheral nervous system, made of the sensory division and the motor division. Now you know what is sensory, meaning you're feeling something coming to you. But where does it come from? If it comes from your internal organs, we call that visceral sensory division of the nervous system. But if the information comes from your muscles, your joints, or your skin, we call that somatic sensory. Now, soma means your body, coming from your body. Let's say you receive information from the visceral or somatic, it goes to your brain, so to the central nervous system. And the central nervous system has to make a decision. If that decision is made and response has to be given, it will be given through the motor division of the nervous system. Now, where does it go to? If the command is given to a muscle to do something, it's called that division, that area of the nervous system is called somatic motor division. But if it's given to an internal organ to do something, your heart beating, your blood vessels shrinking, dilating, or constricting, that is called visceral motor division. But when the response is given to an internal organ to do something, let's say, for example, your blood vessel has to be dilating or constricting, or your heart has to beat, that is what we call visceral motor division. Now, the visceral motor division is also called what? Autonomic nervous system. Why? Because it's done for you automatically. Now, since it's done for you, you don't make a decision. Your brain has to make a decision about what needs to be done. If it's doing something, for example, in the response where you have to kind of take action, that division is called sympathetic division. But if it's a division that allows you to relax and to take slow, it's called the parasympathetic division, nervous system. So you can see here that the nervous system has different branches based on where the stimulus is coming from and where it's going to. Now, why is the peripheral called peripheral? Because it's to the side, right? It's not in the middle. It's not the brain, it's not the spinal cord. It's to the side. Why the central is called central? Because it's central. It's in the center. It's central. It's like where everything is processed. Now, how is your own sensory division help you do what your motor division cannot? Now, your sensory allows you to feel things coming into your body. But the motor division allows you to do something, to respond to something. Why this is important? Because sometimes you can have sensory damage, but not motor damage. It means that if you have sensory damage, you don't feel anything. But the motor will do something. That means you can walk, you can do stuff, but you don't feel it. But if you have motor damage, then you cannot walk, you cannot do something because it's going to be blocking the motor neuron that allows you to do something. So that's why the sensory and motor are very different. One takes in sense, the other makes you do something motor. So what is the difference between somatic motor division and visceral motor division? Now you can see somatic motor division tells your muscle to do something, But your visceral motor division, also called autonomic nervous system, allows your body to take responses automatically inside of your organs that you don't control, divided into sympathetic and parasympathetic nervous system. And the next question is the difference between neurons and nerves and between ganglia and nuclei. Now, what is a neuron? A neuron is a cell. It's a cell, okay? Now, what type of cell is it? It is the cell of the nervous tissue. It is the main cell, the cell that does the action potential, that does the work of the nervous system. You can see the neuron here with the cell body, the nucleus, the dendrite, At the dendrite level, that's where you receive information to be processed and for an action potential to be triggered through the axon to go to the axon terminal. So at the end, there can be some kind of neurotransmitter, some kind of response given to the next neuron or to the next muscle or to the next organ to do something. But what is a nerve? When you take many of these axon terminals of these neurons, you form a nerve. So nerves are just groups of axon terminals of neurons. Now you can tell if I look at this picture on my right, you see these little pieces are the neurons. You put a lot of neurons together, right? Many neurons, you form, because a neuron is a cell, you form a tissue, and the tissue in nerve is called what? Fascicle. Then when you take a lot of fasciculi together, you mix a lot of fasciculi, you form the nerve fiber, which will be what? Like what? An organ, because organs are made of tissues, tissues are made of cells. So the cell is the neuron, the tissue is the fascicle, and the organ is the nerve. Now you can see this is a spinal nerve coming from the spinal cord. It will have a sensory, right? The unipolar sensory taking information to the spinal cord, and the ventral root, which will come to give the command. So you have dorsal root, that's why you have the dorsal root ganglia, which is groups of cell bodies of unipolar neurons. Now here also you can see the gray matter and white matter we talk about. See, the gray matter here is inside, and then the white matter is outside. That's contrary to the brain, okay? So nerve, a cord-like organ. So the nerve is a cord composed of numerous nerve fibers. And these fibers are what? Just axon, bound together by connective tissue, right? So they have both sensory and motor, because the sensory and motor kind of divided here at the rootless level before they get to the spinal cord. Now give the name of the 4 types of neurons based on their anatomical structure and explain the location and function of each type. Now here we got 4 types of neuron based on their shape: multipolar neurons, bipolar, unipolar, and axonic. What's the difference? Anaxonic, they don't have no axon, and negative. Unipolar, you only see one little line coming from the cell body, uni, just one extension from the axon and the dendrite at end. The bipolar, you have 2 extensions from the cell body, and then the multipolar, you have many dendrite and axon coming in different direction. So these are based on the structure. Now, but what about the function? What do they do? Based on function, we have 3 types of neurons. We have sensory neurons that take information, interneuron that process information, and motor neuron that carries out the command given by the interneuron. Now, what do the sensory neurons look like if you go to anatomy? They're going to be unipolar neuron, or they could be bipolar, because we have sensory organs, special sense organs that have bipolar neuron that makes you feel something. So they're going to be sensory. What about the interneuron? You can see here there are multipolar neurons, and the motor neurons here, you can see they're also multipolar neurons. So that's going to be based on their function, what they do. They can either feel, process, or do something. But the shape, you can have 4 different types. What about the glial cell? Give the name of the 4 types of glial cell in the CNS and specify the function of each. Now let's talk about this. Glial cells in the CNS, the acronym is going to be MEOW. That's going to be what? Look at them. Microglia, their job is to clean up around the neuron. Ependymal cell, their job is to secrete the cerebrospinal fluid. Astrocyte, here you can see them, their job is to protect the neuron, guide the neuron, nourish the neuron, covers the neuron, some kind of way to give the neuron its shape, its structure, its ability to do its function. And then oligodendrocyte, you can see them here. They covers the neuron. They're the myelin covering of the neuron. They protect the neuron. They're the myelin. They allow action potential to run fast, right? Then when you go to PNS, the peripheral nervous system, we have SS Schwann cell and satellite cell. Satellite cells, they look like astrocyte. So Schwann and satellite are the 2 glial cells in the peripheral nervous system. Why do we need covering of the neuron? Why do we need myelin covering? Because it increases the speed of action potential, and which depends on 2 things: The diameter of the fiber, the bigger the fibers are, nerve fibers are, the faster the action potential. Things run fast in big nerve. And the presence of myelin. When you have myelin covering on the neuron, the action potential will be fast. Now, what structure makes the white matter white and make the gray matter gray? The white matter is white because of the myelin. And now we know myelin in the CNS, it is oligodendrocyte. In the PNS, it's Schwann cell. What makes the gray matter gray? It is made of Nissl body. What is Nissl body? That's going to be like the endoplasmic reticulum of the neuron, right? Because those are proteins. They look dark. They're in the cell body of the neuron. So the cell body of the neuron make the gray matter gray because they have inside of them Nissl body. Let's go to the next set of questions, 13 to 20. Copy this graph. So this graph is the graph showing the action potential. It has numbers. You can see number 1, local potential, number 2, threshold, then number 3, depolarization, number 4, plus 35, number 5, repolarization, number 6, hyperpolarization, and number 7, coming back to the resting membrane potential again. What is this? This is just the graph to show you how the inside of the cell of the neuron changes from negative 70 millivolt to plus 35 to come back to itself. This graph is just showing how action potential happens, which is such a movement of ions inside the neuron, in and out, depending on the situation. So we said we need to use all these keywords when we explain this. You must list the 5 keywords. What I can use here is that at the resting membrane potential at negative 70 millivolts, the cell is at rest. Then, when local potentials happen, that means the cell is excited a little tiny bit with something, sodium will move into the cell to make the cell less negative, from negative 70 to negative 55. When you get to negative 55, it reaches threshold. The potential reaches the threshold. What does that mean? It is the minimum level that allow an action potential to happen. But what happens then? It is because sodium, which is positive, moves into the cell to make the cell less positive. At this threshold, negative 55, so a voltage-gated channel, what does that mean? It just means there are channels that open when the voltage changes, when the light changes, when the lights come on, basically. When they open to allow even more sodium to come in to make the cell less negative up to 0, even positive to plus 35. This movement from negative 70 to plus 35 is called the depolarization. Why? Because remember the cell used to be negative inside and positive outside. Now it's positive inside and positive outside. There's no pole. There's no plus and minus. It is depolarized. You remove the pole. Then when it gets to 35 plus 35, the sodium door, which is the sodium gates, will close and the potassium gates open. Because the potassium is very, very high concentrated inside the cell, when you open, the natural will leak out. They will come out. When they go out, because potassium is positive, when something positive comes out of the cell, it makes the cell negative again. Coming back again to where it used to be is called repolarization. Repolarization. This whole process is called an action potential. But at the bottom here, number 6 means when it gets back to negative 70, the potassium gate, which is just the potassium door, will close. While they're closing, there's still more potassium coming out, which will be making the cell even less negative than the normal resting membrane potential. We call that hyperpolarization. So the cell will come to even less than negative 70 and then come back to itself. So here we need to know how to do this graph. So now when you look at this graph, you should look at the A and say, okay, negative 70, and then B, graded potential or local potential, C, depolarization, D, repolarization, E, hyperpolarization, and F, resting membrane potential. Now, here we see all the names here. What is negative 90 millivolts? It is hyperpolarization. Now here, we just show you at the bottom how the graded potential, the local potential are formed at the dendrite, and they add up to become an action potential all the way down to the end of axon. What happens at the local potential is just some sodium moving in to excite the voltage gated sodium channel that need to be open. Now, why we talk about sodium and potassium? It is because when things happen in the neuron, that thing is called action potential. It is just the movement of ion. Sodium coming in, potassium going out. When it's at -70, sodium will come in to make the cell less negative. What does less negative means? From -70 to -55 to 0 until it becomes positive. When it becomes positive up to 35, sodium gates closes, potassium gates open. Now when potassium gates open, it will allow potassium to go out to make the cell come back to where it used to be. We call that repolarization, right? So that's what it's explaining here. Now let's explain the difference between continuous propagation and saltatory propagation. What does that mean? It means that during the movement of action potential, if there's no myelin on the neuron, the action potential will be continuously from point A to point B. That is going to be a slow movement. But in saltatory, when there is a myelin sheath covering the neuron, the action potential will jump from node to node. Remember, these are nodes of Ranvier, and the myelin area is called internode. From node to node to node is called saltatory. It kind of jumps from node to node. It's kind of faster. So saltatory is faster, that continues. Then here, I'm going to go back to question 17. Why sodium has higher electrochemical gradient than potassium during an action potential? Let me go back here and explain. Here you see sodium is positive. You have a lot of sodium outside. But the cell inside is negative. If you're positive outside, you want to go inside if inside is negative, because plus positive and negative attract, right? Opposite attract. But if you think about potassium, potassium is positive and the cell inside is negative. So potassium wants to stay inside. It doesn't want to come out. That's why sodium feel like coming into the cell more than potassium feel like going out. But guess what? At the same time, the gates, the door of potassium are more permeable. They allow more potassium to go out. That's why, despite the high electrochemical gradient of sodium, when potassium gates open, potassium will go out because sodium cannot use that door, right? So here, we need to understand why potassium affects more the action potential, because potassium is more concentrated in the cell. At the same time, the potassium gates open faster. They open more potassium gates. That's why the resting potential depends on potassium more than it depends on sodium. Even though it is the sodium that moves in first, because the sodium is positive, it wants to go into the negative area. Now, question 16 says, what determines whether an action potential can be initiated in the postsynaptic neuron when they are both inhibitory and excitatory? Let me explain what is inhibitory excitatory. Now, when the neuron talks to another neuron, we call the form a synapse. The neuron connecting to the next neuron, the first neuron is called the presynaptic and the second one is called the postsynaptic. Depending on the neurotransmitter that's being secreted, that neuron can make the next neuron excited or it can stop it from even working. It's called inhibitory. Why this is important? Because every neuron will respond to both of them, excitatory and inhibitory, because both determines the value, the end effect of the action. So that's why it is not only inhibitory or only excitatory, it's the combination of all the information coming to the neuron to make the neuron work. Now let's explain the difference between an action potential and the graded potential. We saw that an action potential is made of depolarization, sodium coming in, repolarization, potassium going out. What about the graded or local potential? Before an action potential can form, small potential have to add up together to form a big potential. Those small potential are called graded potential or local potential. Now, they can vary in magnitude. It means that in action potential, for example, you can either have it or not. It's called a rule of all or none response. Either it happens or it doesn't happen in the action potential. But in the graded potential, it can happen a little bit. So you can have different magnitude of the stimulus. That's the first difference between graded potential and action potential. And then the graded potential is decremental, meaning that if the graded potential runs through the neuron, it'll get smaller as it goes to the end. So the voltage shift caused by sodium inflow diminishes with the distance. As it goes, the power reduces. In action potential, it doesn't reduce. It either happens or it doesn't. And the graded potential or local potential is reversible. If you give a stimulus, you will have a graded potential or local potential. But if it stops, it will stop. But in action potential, it's going to be different. Once you run an action potential, it will always happen. Now, a graded potential can be either excitatory or inhibitory. What does excitatory mean? We know that a cell is negative inside, negative 70 millivolt. In excitatory graded potential, the cell becomes less negative. From negative 70, it could go to negative 60, negative 50, and up to plus 35. See, it kind of go up. But before you get to negative 55, we call that graded. Once you hit the threshold, whatever comes after that is called an action potential. So the graded potential can run from negative 70 up to a threshold. And then from that threshold, you're going to hit an action potential, which will either happen or it doesn't happen. Or the graded potential can also be inhibitory. What does that mean? That means there's some neurotransmitter whose function of the neuron makes the neuron less likely, not more, but less likely to fire an action potential. We call that inhibitory. For example, gamma aminobutyric acid, is a neurotransmitter that kind of makes some cells less likely to fire action potential. Why? Because they're going to take the influx of chlorine inside of them, which will make the cell even more negative, from negative 70 to negative 90. So to reach back the threshold, it'll take more time and energy. That's why these are the 5 things we need to know about the graded potential, about local potential, right? It's the same thing, graded or local. In action potential, we saw the graph. We know either it happens or it doesn't happen. So to summarize this lecture, this is a review of the nervous system, right? We learned about the function of the nervous system, how it's divided, the brain and the spinal cord, the differences, and then we also learned about the action potential, the graded potential, the neuroglia and the neuron, so that we can see the relationship between the shape of the neuron and its function, the presence of myelin and its effect on the speed of the action potential, the difference between a full-blown action potential and localized graded potential that build up to become an action potential. All right, thank you very much, and we'll talk about the next step in the next video. Thank you.